So the next thing we're going to talk about is essentially how our lymphocytes become activated. So our lymphocytes, okay, go through a stepwise process of essentially being able to carry out the functions that they carry out. But before they can do that, they have to go through a very, very rigorous process, okay, that involves 5 major steps. So kind of our first step is the fact that our lymphocytes are where they originate from, okay? So kind of the first thing is knowing that both our B cells and our T cells originate in the red bone marrow, okay? And you can think about this, right? When we think about origin, where they're born, okay? So our B's and T cells are born in bone marrow. Once they're created, okay, they then go off and undergo a process known as maturation. On the next number of slides, we're going to go over this process of maturation, which is basically a process of them becoming educated. They then go on and they seed. We'll talk about what they're seeding. Here they're going to encounter, at some point after they've seeded, an antigen and become activated. And then finally, to be actually fully activated and carry out the fight of the pathogen, they're going to undergo proliferation and differentiation. Proliferation just means division, cell division. Okay. And differentiation means that they are going to transform into something slightly different. So let's start with maturation. Again, both our B cells and our T cells are born in the bone marrow. After they essentially are made in the bone marrow, okay, our B cells stay in the bone marrow to become educated. Our T cells leave the bone marrow and essentially populate the thymus. And in the thymus is where they are going to become educated. Now, this education process will create lymphocytes that are capable of doing 2 things. One, it'll create a lymphocyte that is able to basically demonstrates immunocompetence. And immunocompetence basically is a lymphocyte's ability to recognize one specific antigen by binding to it. The key here, remember I mentioned to you guys in the previous video that a B cell can only ever recognize a single antigenic determinant, okay, which means it can only recognize one antigen, and on that antigen, one antigenic determinant, okay? So, once a B and T cell reaches maturity, okay, they will display unique receptors on their surface, okay, which can only bind one antigen. Okay. The other thing that the B cells and T cells acquire upon graduating is something called self-tolerance. This means a lymphocyte is unresponsive to its own antigens. Okay. Now, we're going to look at this process of maturation in T cells. The process of maturation is better understood in how T cells undergo this education process, but we're going to assume that the maturation process for T cells and B cells is identical. Okay, so even though on the next slide I'm talking about the T cells and their maturation process or their education process, we're going to assume the B cells do it in the exact same fashion. So, we already said that the T cells will undergo this education in the thymus. Now, just like you guys have to take tests, T cells and B cells also have to take tests. Okay? So, I'm going to go back and we're going to kind of draw a cell. Okay? So this is a T cell. And we're going to call it a baby T cell. Uneducated. Okay. Now, this T cell, because it's our cell, right, is going to have markers on it. Okay. Okay. Let's not do this. Let's make this a different color. These are going to have markers on them. Oh, come on, color. Okay. Now, this is a receptor. Right? We just said up here they have unique receptors on them. Okay. Now, in this selection process, we're also going to have an antigen presenting cell. And we're going to call this an APC. Now, an antigen presenting cell has on its surface a marker that indicates it as self, right? So this is our MHC, okay, protein. And again, this is identifying the cell belongs to self. Okay. And on that MHC, right, we have a self antigen. Okay. So this is kind of what I drew last time, last lecture. Okay. So we need, here's our baby cell. We need this T cell to become educated to the cells of our body. So we want, okay, just dang it, that T cell, baby, okay, we want it to recognize this MHC protein because we need it to know how to identify or how to recognize a cell that has a self marker, right? So we need it to identify this. Okay. We don't want it to recognize the self antigen, right? If this recognizes this MHC, which is good, we want it to recognize that, but also recognizes the self antigen, then we're going to elicit an immune response. So we need to train our Bs and our T cells in this maturation process to recognize the MHC, but to not recognize the self antigen. Okay. So what this creates is two tests. A positive selection, okay, because we want something to happen, and a negative selection, which is we don't want to recognize the self-antigen, okay? So essentially I just wanted to draw it out so I could explain it, but this picture shows you exactly what that is. Okay, so here's our positive selection. So if we go back, let's look at this. Okay, here's our baby cell. We want it to recognize this MHC molecule. Okay, so what'll happen is here's our baby T cell. Here's its receptor. Okay, we want this receptor here to recognize this here. Okay. Now you can see here, it doesn't recognize that. Okay. However, here, it does. Okay. So sad face up here, happy face down here. Okay. So again, that's the positive process, positive selection. We want something positive is something good. We recognize something. We know something. Okay. So in the case where we match, think of a conveyor belt, right? They match, it gets to survive. It doesn't match, it undergoes something called apoptosis, which is death by cell suicide. Okay? So, in positive selection, if the MHC, if the receptor of the T cell does not recognize the MHC molecule, it dies. We want it to recognize, because again, remember, the MHC molecule is what identifies this cell as belonging to self. Okay? Now, our negative selection is that if we go back, okay, so let's look, negative selection, we don't want to recognize the self-antigen. Okay? So, here we are down here. Okay, now what we're referring to is this little orange thing. We don't want to recognize that. So, if we don't recognize the self-antigen, we survive. However, if we do, so this is happy face, if we do recognize this self-antigen, which we see here, perfect fit, again, we undergo apoptosis. Okay. Now, this will take a little bit of conceptually drawing out and going through, okay? But hopefully the drawing on the previous slide will help us. Ultimately, what this process does, it ensures self-tolerance. That negative selection ensures that when that T cell and that B cell graduate, okay, that they've acquired self-tolerance, okay, which is the inability or the unresponsiveness to its own antigens. Okay, so that was our step 2 in our step process. Okay. Step 3, okay, our baby is now graduated. Oh my gosh, it's so exciting, okay? So now they've been in the little police academy this whole time, learning who the good guy is and who the bad guy is, or not even learning that, just learning the basics. We don't even know who the good guy and the bad guy is yet. We just know we've like armed them with the tools to be able to distinguish the good guys from the bad guys, okay? So what happens is those cells leave the bone marrow and leave the thymus and are educated, okay? They've gone through the tests. They know what they're supposed to be doing, okay? But they're still naive, which means they've never encountered a pathogen or an antigen yet, a foreign antigen, okay? So what they do is they go out, now that they're educated, they're the rookies, and they're going to seed our secondary lymphoid organs. Remember, the bone marrow and the thymus are our primary lymphoid organs. And what we mean by seed is they're going to go and populate our secondary lymphoid organs and circulate within our blood and our lymph. And what they're going to be doing, right, they're going to be those patrol officers on the overpass just watching everything go by. And when they recognize something that's not supposed to be there, they'll undergo antigen encounter and activation. Okay. Now, again, we're going to assume that maturation process we just went through, both B cells and T cells. What your book does from here on out is it focuses on B cells, and then it comes back and talks about T cells. Okay. In the video that I provided, which talks about proliferation and differentiation, a drawing that I did, I start with T cells and then talk about B cells because to me it makes more sense to talk about T cells first. But I'm going to follow the book's lead on this one for the lecture purposes. Okay? So, we have a naive lymphocyte. Okay, again, rookie patrolwoman out in our secondary lymphoid organs. Okay? And all of a sudden, it encounters an antigen. Okay? When that encounter occurs, that means that cell is going to trigger further development. And we get something called clonal selection. Okay? So, essentially, again, we're going to talk about B cells first. Okay? So, when we recognize or when we essentially encounter that antigen, okay, when we talk about activation, what's going to happen is that cell, that naive cell, will undergo proliferation and differentiation. Okay? And it's a stepwise process. So the first thing that's going to happen is our B cells will become activated when that antigen binds to the surface receptor and you basically get a cross-link. Okay? The receptor, through a receptor-mediated endocytosis process, okay, will basically internalize that antigen. And one of two things will happen. That cell will differentiate or will divide, of which will become either a effector cell. I always call them plasma cells. Okay? And plasma cells are going to produce antibodies. Okay? Now this is very specific to B cell differentiation, proliferation differentiation. Okay? These plasma cells will produce antibodies, they'll survive, they'll exist in circulation for about 4 to 5 days and then they die. However, when that cell differentiates or proliferates, which is dividing and then it differentiates, it will also form memory B cell. And that B cell will always remain or retain immunological memory, which means the next time that it encounters a pathogen, okay, that same pathogen or antigen, it can elicit a response much, much faster. Okay. Okay, so let's look and see how this process works. We have, okay, our antigen, the little red thing, little red circles, entered our body. Okay. And we have a receptor on the surface of our B cell. Okay. Now, what you can see is only one of these B cells is actually responding because it's the only one that has a receptor that's complementary to the antigen. Okay. So these guys don't respond because they aren't meant to recognize this antigen. However, this antigen binds to this receptor on our B cell, okay, activates it. We get proliferation. So this cell becomes 2 cells, and these 2 cells become 4 cells, and 4 cells become 8, and 8 becomes 16, and so on and so forth. Okay? And then these B cells, these activated B cells, undergo differentiation. So from here to here is differentiation, where the majority of the cells become plasma cells, and a small portion of them become memory cells. Now, the reason the majority of them become plasma cells is the plasma cells are the ones that are going to be fighting, right? If we have an infection, we don't really care about memory if we can't fight the current infection because it's going to kill us. Okay? So what we're focused on is the current infection. So the majority of the cells that are going to differentiate are going to be plasma cells. Okay? So the importance of those memory cells, however, come back into play when we're re-exposed to the same antigen. Okay, so let's look at kind of what we mean here. I like this little chart over here because it really gives you a really good visual for what we're talking about, okay? So you know how we talked about the fact that the adaptive immune system has to be primed, okay? So you're exposed to a pathogen on day 0, a pathogen you've never seen before, okay? It takes about 5 days for that process we just talked about to occur, okay? So it's about 5 days before your body even starts to produce its first antibody. Crazy. Okay? And we differentiate, we produce our plasma cells, it fights the infection, it releases all of these antibodies, right? Meanwhile, the infection is dissipating, the B cells are dying, okay? And we resolve the infection. Okay? Now, a year later, you're exposed to the exact same pathogen, okay? And this is represented here by day 28. Now, unlike the first time you were exposed to it, the second time, you don't have to prime. Within hours, okay, you're producing antibodies. And not only are you producing antibodies, within a few days, you're producing I don't even know how many times more antibodies than you did the first time around. Okay? So these antibody levels can actually remain high for months to weeks after an infection. Okay? Especially these ones here, to kind of combat that infection from trying to come back again. Okay? Now, when you're sick, right? The reason you're sick is usually within this 5-day window. Upon second exposure of the same antigen, you rarely know you're sick or know that you've encountered a pathogen because your body's able to combat it so quickly. Okay. So that's the significance of that immunological memory. Okay. Now, when we talk about immunity, okay, that immunological memory, that ability to generate an immune response, okay, there's 2 types of humoral immunity that exist. There's active immunity and there's passive immunity, okay? Now, active immunity, okay, in active immunity, you're going to generate memory. Okay? And that is because one of 2 things have happened. You've either naturally acquired a pathogen, i.e., you touch a door handle and then you lick your fingers. Okay? That's naturally acquiring. And the reason it's considered active is because we're going to go through all those steps we just went through. So your body is going to see that antigen. You're going to elicit the adaptive immune system. That B cell is going to activate, undergo proliferation, differentiation, and going to generate memory. Okay? However, you can also generate memory artificially. Okay? For example, I could give someone a vaccine. Okay? In that vaccine contains the antigen. Now, most vaccines, the pathogenic component of that vaccine or that pathogen has been removed. So the disease-causing portion of the pathogen has been removed, and all the body is seeing is fragments of that pathogen. Okay? So, because you're acquiring it artificially through a vaccine, through an injection, okay, we call it artificially acquired. However, because you're still seeing the antigen, you're going to generate memory. Okay? So again, we generate memory because we see the antigen. Okay? Now, there's also passive humoral immunity. And in passive humoral immunity, okay, this is transient or short-lived, okay? And the reason for that is there's no memory established, okay, because not seeing the antigen, okay? So let's look at these two processes. Again, we can acquire passive humoral immunity naturally. And in this case, what we are acquiring is the antibody, okay? So the body is never seeing the pathogen. We're never going through that proliferation, differentiation process to generate memory. Instead, we are being given the antibody. Now, naturally, this can occur. One of the classic is breast milk, okay? In breast milk, we have IgA antibodies, okay, that help prevent entry of pathogens into the bloodstream or into the body of the infant. The placenta, okay, to the fetus is also another one. So in that case, we're not giving the infant the cells or the memory or the pathogen or the antigen. We're just giving them the antibody. So as long as you're breastfeeding, okay, that baby will have that protection. The minute she stops, that antibody is gone. No more protection. Okay? The other way we can acquire passive humoral immunity, again, is through an artificial mechanism. So again, this is like an injection. However, in this case, again, we're injecting antibodies. Okay? Not an antigen. So again, this antibody works for a short period of time. Eventually, over time, it's going to break down because we know antibodies can be there for a few months, for a few weeks to a few months. Okay? And once it's broken down, there's no memory there to reestablish or to elicit a memory response. Okay? So we talked a lot about these antibodies. Okay? Now, here is what an antibody looks like. I always draw antibodies like this, okay? And that is because they actually do have a Y structure to them. Kind of like that, okay? Now, do not memorize the structure. You don't need to know the heavy chains and the light chains or the hinge regions and what makes different antibodies, okay? But essentially, antibodies are what we call immunoglobulins, okay? And they're proteins that are secreted by our plasma cells. Okay, so these are, remember, our plasma cells are activated B cells. Okay, and they're capable of binding specifically with antigens detected by that B cell. Now, there's 5 classes of Ig or immunoglobulin antibodies. We have IgMs, IgAs, IgDs, IgEs, RGs, and IgEs, okay? Now, these different classes all kind of have different functions. So, for example, IgMs are the first antibody released, okay? They're really good at agglutinating, and we'll talk about that here on the next few slides. And they're going to essentially activate complement, okay? Don't worry about this word here. I think readily fixes kind of throws students off. But essentially it activates the complement, right? That system that's part of the antimicrobial system, that's part of the innate defenses. IgAs are secreted within our mucus and other secretions. So for example, this is the one I told you, it's found in breast milk, okay? This helps prevent entry of pathogens into the body itself. IgDs are a type of antibody that's actually functions as a receptor on our B cells, okay? IgGs, these are secreted and in the majority of the antibodies circulating, we'll typically find IgGs in secondary and late primary responses. So if an infection's been around or you've seen the infection before, you'll see a whole bunch of IgGs. And IgEs are active in allergic and parasitic infections. And these are the culprits that cause our mast cells and our basophils to release histamine during like an anaphylactic reaction. So when we think about the function of antibodies and why they're so critical, okay, antibodies will inactivate and tag antigens. They do not destroy them. The antibodies themselves do not destroy them. However, when an antibody binds to an antigen, it forms this antigen-antibody complex, okay? And it's this complex that then is kind of tagged for destruction. And it does this through 4 mechanisms. And they're stated here, but I'm a visual person. I like the drawings, okay? So here's our antigen, here is our antibody, and these are going to bind to each other, okay? So they can do one of 2 things, okay? They are going to neutralize. Essentially, here's our pathogen, okay? This antibody will coat the pathogen, okay? And when it coats that pathogen, it targets it for phagocytosis by our innate defenses, okay? It can agglutinate, which means these antibodies, right, bind to the antigens on the red blood cell, and then these antibodies actually clump together, and this clumping here also triggers phagocytosis. And the last one is precipitation. So these antigen-antibody complexes can actually come together and form a much larger mass. This larger mass becomes a signal for our phagocytic cells come along and phagocytize. So all 3 of these kind of consequences of antigen-antibody complexes will trigger phagocytosis. The other thing that this antigen-antibody complex will do is activate complement. Remember that series of events. So we have opsonization. Again, this was talked about in video 1. We have increased inflammation, which we see over here, right? And we have the MAC molecule. So here that MAC molecule gets inserted and our cell lyses, okay, and kills that pathogen, okay? So this is how our antibodies actually function. They in themselves do not destroy, but they mark things so that destruction can occur, okay?